Okay, thank you, Avery. And thank you, John, for that. It was really interesting to go back down memory lane to see where it all started, and I couldn't have said it better myself. So it's been a pleasure working with you and everyone else. So thank you for that. So again, my name is Max, and I'll be sharing my research titled Densities of Understory Macroalgae and Substrate Classification in Southern Hood Canal. Okay, so we live in a very rich and diverse region when it comes to kelp. There are about 22 species of kelp in Puget Sound, of which 20 are classified as understory kelp. And so that's really what got me thinking that I would really like to look further into this to see if we can't clear up some potential gaps in understanding. So when I talk about understory kelp, for this research, I'm talking about prostrate kelps, which lack that rigid stiper stem that keeps it suspended in the water column. So if you look at that figure in the middle, you'll see the prostrate kelps are kind of horizontal on the seafloor, and that's kind of what we were looking at for this research. And they are critical in kelp forest communities because it's similar to a terrestrial forest where it offers that multi-layered vertical habitat structure. And so that's what really got me interested in studying them. So again, to rephrase that, understory kelp provide their habitat forming species and provide that living habitat, which provides critical infrastructure for a diverse variety of marine organisms. And their significantly high rates of photosynthesis fuel both marine and terrestrial food webs. For instance, they feed commercially important filter feeders like clams, oysters, and mussels, which in turn benefit our communities. Preliminary aerial imagery, that image you see there on the screen was from an aerial imagery project looking at nearshore communities. And so from that, on top of multiple other variables like substrate and shoreline geomorphology, they found that understory kelps are actually nearly almost 3 times as common as the canopy kelp, such as bull kelp in Puget Sound. So in that case, I was looking to collect data through site-specific surveys, so at a smaller scale, and contribute a small-scale data set for future research and conservation efforts. So to do that, I have a few research questions here. Number 1, what are the densities of understory kelp and percent cover of other macroalgae at 2 different sites in Southern Hood Canal? Are they significantly different between 2 sites? And what are the primary and secondary substrate types at each site to better characterize the sites? And does substrate type and cover vary significantly between those sites? So looking at the sites from a zoomed out perspective, we see them at the really southern extent of Hood Canal, just because that's where I got my experience scuba diving, and I had some experience at those sites and some preliminary observations. I noticed that there's quite a bit of understory kelp here. So you'll see the yellow house is where I work. It's an Airbnb, like a lodge for scuba divers. And so it has access to a little cove. And then Sundrock, a few miles north of there, is a well-known spot for scuba divers. It's a conservation area. So I wanted to go ahead and compare Yellow House Cove to Sundrock. So that's, you'll see site A and site B repeated throughout this presentation. So zooming in a little bit, we see site A and site B, that kind of survey layout, the design here. So essentially we have 5 transects at each site with 6 square meter quadrats on each transect. Those are the little red squares. Sorry if it's hard to see, but we began from a randomly selected starting point, a point of interest that is actually shown on those maps, the coordinates, and 30 samples altogether at each site for a total population sample size of 60. And the methods here, we use scuba surveys, thankfully. And we began at depths of 3 meters relative to mean lower low water. So we made tidal corrections to make sure that we were consistent across the surveys. And one of my dive buddy there that you see on the bottom left, deployed a 30-meter open reel transect along the sea floor, perpendicular to the shoreline and moving directly east as best we could, because that's the shoreline orientation made that easy in that sense. So myself, I followed along with the 1 square meter PVC quadrat and placed them at 5-meter intervals for, again, 6 quadrat placements per transect. And To repeat this process, each transect was located 5 meters north. So we did some, we used compass headings to navigate our way underwater. And so those were 5 meters north of the starting point of the each previous transect. So my duty, so here are the steps in which I took to get these measurements. So first, I would look for any individual kelp types. That's the stem that I could count out. So actual densities, numbers of individuals per square meter, and a visual estimate of macroalgae, so any other algae like red, green algae, as well as the invasive sargassum that you can see there on the right, using methods developed by the Marine Plan Partnership of the North Pacific Coast. And substrate was basically the last thing I looked at during each quadrat placement. So looking at substrate type like boulder, cobble, pebble, and sand, so large grain size to small grain size, as well as their cover level, which was just 3 different type of differentiating factors such as dominant, non-dominant, non-dominant being anything less than 50% of the quadrat. And so even though this is mostly like preliminary data collection, I also wanted to get an angle of statistical analysis here. So using RStudio, I used a non-parametric 2-sample t-test called the Mann-Whitney U-test for kelp densities. For macroalgae, I used a Kruskal-Wallis H-test, which is also a non-parametric test that compares the ranked means across both sites. And then finally, for substrate, a chi-square analysis or a contingency analysis, looking at the variables of substrate type and its association with cover level. And then finally, substrate type versus macroalgae cover, just get that extra layer of quality and data. So beginning with the results here, we have the kelp densities. 2 species of kelp were observed in the study, Saccharina latissima, sugar kelp, and Neoagarum fimbriatum, which is fringed sieve kelp. And they're both large brown macroalgae kelps. And as you can see here, their densities are plotted against depth for each site, depth shown in a dotted line in meters. And so mean densities and standard deviations are shown below each graph. So site A had significantly higher abundances of both species. And the highest density, however, was observed at Sunrock, site B, at 15 individuals in 1 square meter quadrat. So however, as you can see, the kelp at site B were pretty restricted to deeper depths there. We tried to standardize it across both sites, but of course the bottom contour of Sunrock is quite steep. And so it was interesting to see the spatial or the depth distributions across these sites. So there are the mean densities, 3.8 individuals for S. latissima, 2.8 of N. fimbriatum at site A, 2.4 per square meter for S. latissima at site B, and 1 individual per square meter for N. fimbriatum at site B, with their standard deviations as well. Also, kelp densities using that Mann-Whitney U-test, they both varied significantly between sites. S. latissima, you can see that right skew at site B, so that 15 individuals is the highest that we got, and site A was more consistent with observations of kelp. Higher mean densities, and N. fimbriatum, although lower, was still pretty prevalent at site A especially. So it was interesting to see those overlapping kelp, understory kelp populations at these sites. Now getting into macroalgae, this was very interesting. Of course, to no surprise, red and green macroalgae were quite common throughout the entire study. Especially green algae was at the highest cover class. You can see here 4, which is high cover from about 60 to 80% cover. So that's pretty significant. And also sargassum was more prevalent at site B, which even though it is a marine protected preserve, I initially would have thought it might be, less so, but we'll get into the substrate and how that might come into play here. So for macroalgae, we have the Kruskal-Wallis, the H values. So those are the test statistics there. You can see all 3 types of macroalgae were significantly different between sites. And again, definitely keeping an eye on sargassum there. It was not too surprising to see higher cover levels or more consistent cover levels of both green and red algae. They're pretty inconspicuous and quite common throughout all of Puget Sound. So for the final type of characterizing variable here, we looked at substrate type and cover. So these are mosaic plots. that show primary and secondary cover of the 4 substrate groups, boulder, cobble, pebble, and sand. Site A on the left there, you can see sand in kind of the salmon color, very, very dominant at site A with smaller grain size. So yeah, primarily sandy with significant secondary cover. So the non-dominant cover there of all the other substrate types except boulder. Boulder was completely absent at site A, shown there in the green. For site B at Sundrock Marine Preserve, you see a larger grain size such as boulder, which comes in as a dominant substrate type for a little bit of the time, but mostly as a non-dominant substrate type. So less than 50% of each quadrat. And you could see the larger grain size overall with that cobble substrate, but an even distribution of all other substrate types as secondary cover as well. So a little bit less sandy substrate, more larger grain sizes there. And the corresponding chi-square analysis found that substrate type and cover level were significantly different between sites. You could see the X-squared test statistic and the extremely low p-value there, which was really no surprise there. Quite different sites with a lot of diversity. So finally, the substrate and macroalgae. This was another type of test that was run just to get that. This was done last among all of them. So these are contingency tables that were basically exported into R. And what that does is it looks at the difference between the observed values and the expected values. So how close are they? And if they are kind of way different from each other, then we might see some association. So doing that, you can see from top left to bottom right and basically color-coded green algae, red algae, and sargassum. Red algae and green algae were not found to have a association with substrate type, which is also not very surprising because you'll find them in very sandy bottoms, but also in larger grain size. However, sargassum tends to prefer larger grain sizes, which is consistent with what in their home range in waters off the coast of Japan, you'll see them with like large grain size like boulder and cobble. So that was interesting to see that a relationship was detected. However, it doesn't tell you like how strong or in what direction. So this is kind of requires more testing. Now into the discussion here, this test using the scuba surveys provides high resolution data on species abundance on a small site specific level. And so of course, we can only capture as much data as 2 divers can allow, but this was basically one of the first times that such a scuba survey was done in this in the Southern Hood Canal, and it was really rewarding to find pretty significant populations of both Saccharina latissima and Neogarum fimbriatum, and they're known to inhabit wave-sheltered sites, which Southern Hood Canal is for sure. And the higher densities of sugar kelp S. latissima is consistent with previous work done in Puget Sound. And like previously mentioned, the differences in depth distributions could have some implications with the kelp. At site B, the kelp were limited to deeper depths between 8 and 10 meters versus at site A, we basically found them through a lot of the depth distribution. And no kelp were observed above 6.1 meters at site B. And sargassum, as you can see in that picture there, that's very high cover, either level 4 or 5 with that boulder substrate that was mentioned earlier. So this has quite a few implications, and I would like to return to the site, or if other environmental management groups or researchers would like to participate in a study that finds how sargassum removal might affect native kelp populations. So, of course, using the scuba surveys has a lot of inherent limitations. There were some roadblocks like John mentioned, but this is just a snapshot survey. It does not reflect seasonal variability. That requires a lot more time and maybe hands on deck, so to say, to see what kind of like demographic information these kelp, so like growth, mortality, and also environmental variables were not measured. So light, salinity, and temperature are all factors that are known to impact kelp populations. So that's something that I would really like to look at in the future as far as understory kelp populations go in Puget Sound. Now to conclude understory kelp and other macroalgae, in a recent survey done by DNR's Marine Vegetation Survey that suggests understory kelp are significantly abundant throughout Puget Sound, which is quite an amazing thing to learn about. And there's a lot of work that's coming into this. So in terms of overall cover, it's very similar to eelgrass. Although, you know, you hear a lot about eelgrass, it's an incredibly important habitat type here in Puget Sound. So more data is needed to quantify those changes in abundance and distribution over time as far as understory kelp. I know there's ROV surveys being done that can provide similar high resolution data, but if possible, scuba surveys are great at getting this up close and personal high resolution data that we need to track changes in abundance at the species level. So again, the significance of this work, this is preliminary data, first time using scuba surveys at these two sites. Sunrock Marine Preserve is an excellent and really rich marine ecosystem. So I would like to do more work there, as well as compare it with maybe other conservation areas, protected areas, to do a large scale it up survey. So moving forward, I highly suggest doing more what MAP calls, the Marine Plan Partnership calls tier 2 surveys, such as species identification, substrate characteristics, and kelp densities. This should remain a high priority, not just for researchers, but environmental management agencies to kind of get a better grasp of what's down there. Yeah, higher resolution data. So I just want to thank not just my friends and family, my parents for working so hard in life to get me here. Faculty members at Evergreen, you've been so invaluable to me. And again, John Kirkpatrick, thank you for being there throughout the way and fighting for me, vouching for me, as well as other faculty like Aaron Avery and Kevin. Thank you. Dive buddy Alex, he's not here today, but thank you. You're out there somewhere for your participation in this underwater data collection. And the Puget Sound Estuarium weren't for you. So you'll see this at some point. So thank you for making it all possible. I'll open the floor to some questions, and thank you for listening and watching.